Good evening, students. I hope you have liked my previous videos which I gave for the students, and this is the best phase of life to study, to learn, to explore, and the LMR now of the next lesson, which is a very important lesson, whether it is CBSE or ICSE. Now the name of the lesson is genetics. Before this, if you have seen the last video of mine, that was related with cell cycle, chromosomes and cell division. This lesson again is related with that same lesson because it is all the nucleus which is doing this. But the things which you are going to read in this lesson and the things which you have in your mind or a mental mapping of the lesson will be according to the order which I have written here. First, you should know what is heredity, the definition of heredity. Then variation. What is variation? How does variation comes? Why sexual reproduction has more variation as compared to asexual reproduction? Then you will study in this lesson about chromosomes. The structure of chromosome was done in the previous lesson. You recap. Chromosomes made up of the sister chromatids. Chromatids are attached at the central point, centromere, and the function of centromere. Now this chromosome is very important, which is going to play a role here. Now here you will study about the types of chromosomes, little bit. The autosomes and the sex chromosomes. Then a term is there in your book called as karyotype. After that, the word genes, which was there in the previous lesson. Now this gene will be in two forms here. Dominant gene and recessive gene. Another important word you will come across will be alleles. After this, you will see certain terms in the lesson. The terms like what is a trait, what is an allele, what is homozygous, what is heterozygous, then phenotype, genotype, dominant trait, recessive trait, monohybrid cross, dihybrid cross, Punett squared, sex-linked inheritance, the sex chromosomes, and in the end will be the contribution of Mendel, who was known as the father of genetics, with his three laws: the law of dominance, the law of segregation, and the law of independent assortment. Okay, now I will not again tell you, I will not teach you the lesson. I will just tell you how you are going to redo the lesson for your board exam, whether ICSE or CBSE. Now first thing here is, you should know what is heredity, and please keep your ICSE book, and if the student is from CBSE, they can keep their NCERT book of their textbook with the textbook prescribed for them, and keep a notebook with you to write what I am telling. First, you should define. The full lesson will be done in form of question answer. This time I will just try to do it. What is heredity? So what will you write for the definition of heredity? Heredity is the transmission of characters. Those characters are resemblances as well as variations from one generation to the next. This you will write for the definition. And this transmission of character is from the parent to the offspring. Okay? Now, this is known as heredity. Second is, what is variation? Now, what is variation? What will you write here? Variation means to change. And the thing which arises from variation is nothing but, it is known as variant. V-A-R-I-A-N-T, variant. And okay, so the definition of variation, which you will write for 1 mark, will be the differences of traits shown by the individual of a species. I repeat, the differences in the traits means character, shown by the individuals of a species and also by the offspring of the same parent is called as variation. This variation is more in case of sexual reproduction as in case of then asexual reproduction. Another important question, why it is more in sexual as compared to asexual? The answer for this is, if you will write or you will think, you will read the book, I give you 4 points here. First is, the chromosomes separate during gamete formation. Second, the crossing over which takes place during meiosis. Third, the chance of coming together of the chromosomes during fertilization because zygote has to be formed. And fourth, sudden change in the genetic order, in the genetic alteration of their DNA, which is called by the word mutation. So these are the 4 points to write, that why sexual reproduction is more important as compared to asexual reproduction. Asexual reproduction produces less variation because it shows it is done by mitosis, if you remember, and sexual is done by meiosis. Meiosis is crossing over chiasmata, recombinant genes, and thus sexual reproduction more important. Okay, this question comes for variation over here. Now, the offsprings which are produced due to variation are known by the word variant, and this variant has to be naturally selected by the environment, and then that variant will evolve, which will lead to the course of evolution, which is not there in your portion. And that was studied by Charles Darwin. That is the natural selection, survival of the fittest. That variant which is more fit to survive will only evolve as compared to the other variant which is not able to survive. Like if there's a hot spring lake where the temperature of that lake is very high, for example, and all bacteria cannot survive there. Only those bacteria which can survive in that high temperature will be able to evolve. And I'm giving you an example, and that will be a variant, and it will evolve. So this is the two things, heredity and variation. Third, I covered that point. What is sexual reproduction? Why it is more important as compared to asexual reproduction. Now, next thing which you will come across in the lesson when you go through is the word chromosome. Chromosome is playing a very important word in your ICSE book, you can see, or your CBSE NCRT book, whichever book. Now, for this, I tell you the things which you should keep in mind for chromosome is, first, you should keep in mind what is a chromosome, where it is found. All this you know, what is a chromosome, where it is found, and what role does it play in reproduction. Okay, what role does it play in reproduction? So for this, the first thing is, please recap the structure of chromosome. The structure of chromosome, chromatids, centromere, that you will write, where it is found, in the nucleus it is found. Now, these chromosomes are of two types for this lesson, and they are called as the autosomes. Autosomes are written here, autosomes are written here, autosomes, and the another one is sex chromosome. Now, for example, the number of chromosomes are constant for a species. Like man, who is called as Homo sapiens. Man has 23 pairs, means 46 chromosomes. 23 pairs means 46 chromosomes. In this 23 pair, we can say, or it is the rule that 1 to 22 are similar. And they are called as autosomes. Say for example, and always these chromosomes exist in pairs. This is a chromosome, this is a chromosome, this is a chromosome, this is a chromosome. They exist in pairs. This is chromosome 1. Like this, till it will be arranged till 23. Okay, till 23. It's given in your book. And this arrangement of chromosomes as per their shape and size is known by the word karyotype. Again they ask, what is karyotype? Arrangement of chromosome as per the shape and size of it is called as a karyotype, in which the first 22 are similar and they are known as autosomes. They control the morphological features, they give the features to your body, all the external features to the body. But the 23rd pair, which is known by the word sex chromosome, which is like this in case of XX, in case of, this is one pair, in case of females, and in case of male it is XY. It's given in your book, and the Y1 is shorter, than this. The 23rd pair is known as the sex chromosome, and it is this sex chromosome where the meiosis takes place, because it is found only in the sperm and the ova. Now, related to this, I will just show you, as I told you, these other terms are there which will be there for your reference. So you should know the definition of these terms before I go for sex chromosome. Okay, so now please quickly write the definitions of, if I give you a question that define the following terms. What the terms which I give you here is, number 1, what will you write for trait? What will you write for trait? First answer, one line answer. You should write, it is an inherited character or that is a feature which is inherited and has its own variant. I repeat again, trait is an inherited character, like the trait for me. I can the trait for tallness, height. That height is a trait, and that it is inherited by that particular next organism, which can be either tall or dwarf. So trait is an inherited character which is normally inherited and has its own variant. Okay, and the traits are also the alternative forms of a character. For example, tallness and dwarfness are the two traits of the character height, as I told you, height. Then comes define the word alleles. What will you write here? The alternative forms or the varieties of genes in a particular gene pair. The diagram is given in your ICSE book, where you can see these alleles are placed on the same different loci of the same gene. The figure number you can please see where the figure is given. It's given on page what is an allele? 1 minute. It's given here. I'll just tell you. You refer to the figure allele, page number 27 ICSE. Alternative forms of a gene occupying the same position on a homologous chromosome and affecting the characteristics but in a different way. Alternative forms of the gene, the different loci of the same chromosome, and these chromosomes are homologous, as I told you. So the alleles are this you will write for alleles. Next thing, if they ask you what is homozygous, so this homozygous is, you can also keep this in mind. Homozygous I've written here this, and heterozygous I've written here this. So what is homozygous and what is heterozygous? For homozygous you should write when a gene pair, this is a gene pair in an organism contains two identical genes controlling the similar trait of a character, then the organism is considered as homozygous. And they are in the same form, PT capital, because it is dominant, and it is the rule that the dominant trait has to be written in capital letters. That is why PT is capital. Okay, this is known as homozygous tallness. If they ask you, if they give you just the symbol in the exam and they ask you write the meaning of the symbol, what will you write? You will write homozygous tallness. It means that. Okay. Now the next one is heterozygous, and the word suggests means different. Now how will you define this word? When a gene pair contains two different genes, capital T, small t, controlling different traits of a character in an organism, it's called as heterozygous. This is PT. Okay, one is dominant, and the one which is written in small letter is also told by the word recessive. There are two words here which will come across, dominant and recessive. Okay, this is another thing. Define these three terms I told you. Alleles, then which one? Homozygous, heterozygous. Now another term here which you should know is what is the phenotype. I think I have written here. No, I have not written. No, I have written phenotype and genotype. Phenotype and genotype. Define phenotype. What will you write here? The physical appearance. That's all. Your hair, the skin color, your that is, for example, phenotype. The genotype, the genetic constitution of an organism. The genetic constitution is known as genotype. And the entire gene and the entire sequence of the DNA is known by the word genome. They also ask what is genome. What is genome? It is the full complement of DNA having all the genes in it for that organism. That is known as genome. Genome. Okay, so these are the 5-6 terms which are very important for this lesson. Next important term you should come across will be F1 generation, F2 generation. F1 means filial, first filial generation, and this generation is produced by the crossing of two parents, male and female, crossbreeding, and we get a hybrid, F1 generation. Next you will come across will be F2 generation, which is known by the word second filial generation, where the individuals are obtained by selfing, that is known as self-pollination, selfing. I will show you in monohybrid and dihybrid cross. Okay, then another word you will come across here you should know is dominant and recessive. Dominant is one which is expresses itself in the first generation, F1 generation. So you will see this. And recessive is one which is very much there, but it is not expressed. It is expressed in the second filial generation. So these are the certain terms which are there for this lesson, which for a mental mapping you should keep in mind. So till now I told you what is heredity, what is variation, what is why sexual reproduction is more important as compared to asexual, the 4 points, and then I came to these terms which you should keep in mind. Also I told you about chromosomes, karyotype, autosomes, sex chromosome. Okay, this was about all this. Now I will just show you sex determination in man. That thing which if they ask you, explain sex determination in man, how will you do this? I will give you one very simple representation over here for just sex determination in man. Please keep your pen and pencil ready. Hope you understand what I am drawing here. The first thing here you should draw is this is diagrammatic representation. If they give the diagrammatic representation of sex determination in man. First you should keep here male into female. This into means fertilization. Now these are the parents. Okay, then comes these parents, the genetic constitution is 44 plus XY, 44 plus XX. This in pairs I said 46 chromosomes. So 44 will be in one set and this is X and Y are the one pair. So that is why total becomes 46. Now then the gametes which they will produce. The gametes produced by them. Second you will write the gametes. What you will write here? It produces half, which you read this in meiosis, meiotic division. 22 plus X. And the half sperm will get 22 plus Y. The male the sex organ is testes. Testes produces sperm and the sperm, half of the sperm will carry X chromosome, half of the sperm will carry Y chromosome. And that is why it is said it is the male which determines the sex of the child, not the mother. Here also what the distribution will be 22 plus XX. Here also it will be 22 plus X. Now you should know one term here. This male chromosome in man is known as heteromorphic. Heteromorphic. H-E-T-E-R-O. Hetero. M-O-R-F-E-H. Heteromorphic. They are different. And the X chromosome in female are homomorphic. They are similar. Homomorphic. Now what will happen in the next step, you see? Here what will happen, these are the sperms and this is the egg. Now what will happen? Now there's one word which is there in your book and this word is not there in CBSE. Punett square. P-U-N-E-T-T. What is a Punett square? What is a Punett square? You can turn to page number 28 if the student is from ICSE and this definition of the book. Punett square is a simple diagram in which the different types of gametes or sex cells with the concerned trait are of one parent like female and the another one is male is arranged in the horizontal way and a vertical way and we fuse them. That is what is a Punett square. They are given there in your book. So I am arranging it in the form of a Punett square. A box which will be divided into 4 parts. One side female, one side male. Okay, so I take here as female 22 plus X, 22 plus X. On this side I take the male 22 plus X, 22 plus Y. Got it? Now I will cross them. I will now cross them, which I told you, cross them. That is this with this. So it becomes this with this means to restore the normal number of chromosomes, meiosis. 44 plus XX. This is a girl. Now this with this. Again 44 plus XX. Again a girl. This with this. 44 plus XY. Boy. This with this. 44 plus XY. Again a sex determination in man. So what do you find here? 50% female, 50% male. Got it? This is the sex determination in man. Rest you can see from your book. What are the sex chromosomes? And what role do they play? And this is what is given here. Got it? How to show sex determination in man. Now we shift to the next part of our LMR. His name the father of genetics. He is known as the father of genetics, and he is nobody else but the Gregor Mendel. He was a monk in Australia, and he wanted to become a teacher, but he could not become. So what he did, but he was a lover of nature. He was very curious to know what is there in the nature, and then he started observing the pea plant. Okay, Gregor Mendel. And what he did when he started observing the pea plant, he observed many things in them. And after that, he started forming some experiments, some postulates, and then he made some laws. The laws are the laws of dominance, the law of independent assortment, the law of segregation. And till date, we follow him, and nobody could replace those laws. So, what did Mendel do? Why he selected the pea plant, and what was his experimental technique, and what did he find out from that pea plant? So, two things which you should always keep in mind is the experimental steps. If they ask you in exam, Mendel, right? Gregor Mendel. The experimental steps. First thing you should keep in mind, what was his experimental steps. Second thing you should keep in mind, why he selected pea plant. Why pea plant? Third thing, what was his observations? What he observed? Observations. Fourth thing, he gave one very important word, and that word was factor. He never knew that this is the genes. Later on, this factor was converted into gene. That he observed that there is a factor which is controlling that particular trait in that organism. Okay, and that he could not tell it as genes, but he just told the word factor, and then he gave the laws. Of genetics. The laws of genetics. Okay, four things. Experimental steps. Why pea plant was selected? What was his observation? What is the factor? What are the laws of genetics? And the observations came under two categories. One was when he observed one character at a time. That is known as mono. So we call it as monohybrid. When two characters at a time. Dihybrid. Okay, monohybrid, dihybrid. Now, students, you will quickly write the question, the LMR for this, the small points which I want to tell you, the experimental steps of Mendel. First, if they ask you, what are the experimental steps? Point 1 which you will write will be the Mendel concluded his breeding experiments in 3 steps. First, he selected a pure parent. Okay, selection of a pure pea plant was taken. That is, the parent or the plant producing similar trait in every generation. Pure. Tallness means tall only, not a mixture of tall and dwarf. So a pure, say the male plant is selected, it will be selected for tallness, and if the female plant will be selected, it will be selected for dwarfness. Capital TT and small t. I will show you next. Then, production of the first generation of plant was done by cross breeding, that is hybridization. Second experimental step. Third, raising of second and the subsequent generation by selfing or self-pollination. 3 things, 3 points. What was his experimental steps? Then, why pea plant was selected? You can see your book, why pea plant was selected. The reasons, if they ask you, give some reasons. The name of the pea plant is Pisum sativum. Okay. It comes in Leguminaceae family. Now, the pea plant had the following characters. It is given in your book. I am repeating again. It is easy to grow. First point. Second, it has a short life cycle, and thus you can study a large number of generations. Third, the pea plant has 7 contrasting characters in them. The 7 contrasting characters are very well shown in your book. And the 7 contrasting characters are like the flower color, dominant trait, purple versus recessive trait, page number 31 for ICSE. Then seed color, yellow versus green. Yellow is dominant, green is recessive. Next is the shape of the seed, round and wrinkled. Round is dominant and wrinkled is recessive. Like that is the pod shape, the pod color, the flower position and the plant height. Figure 3.6, page number 31 for the ICSE students. This is not given for CBSE. So this is very important. I told you Mendel's experiment and why the pea plant was selected with the 7 contrasting characters. Okay. Now, in continuation with what I was telling you about monohybrid cross, where one character was taken, and I had given the example of round and wrinkled and tall and dwarf. And I hope you all can do a Punett square to derive the ratio. The phenotypic ratio was 3:1. Genotypic ratio was also told to you. So that was very important. They can ask you to do in your exam with any set of characters, right? You can see how you have to do it. You have to practice that. Just by seeing, you cannot do. You have to write and draw. Now we come to the dihybrid cross. Dihybrid cross means a cross. If they ask you, define monohybrid and dihybrid. Monohybrid cross, what will you write? A cross where only one character is taken to study. It can be any character, tallness versus height. If you take in height, tall and dwarf, contrasting characters. If you take the texture of the seed, shape of the seed, round or wrinkled. Any one. So we took both the examples and we did it. What is the dihybrid cross definition? The breeding experiments done when we take 2 pairs of contrasting characters. 2 pairs of contrasting characters. I have drawn this here because it will take bit time, so that's why I have drawn it here, and I want you to draw it on your own. You can take 2 set of characters, round and yellow as one parent. Round and yellow you can take as the male parent, one parent, male. You can write here as the male parent. Round and yellow, the male parent, male. And the wrinkled and yellow is the female. So 2 set of parents, round and yellow, male, wrinkled and green, the female. Now, what you see here, see the way they have been written. You have to be very particular with the way you write the alphabets. If you write it wrong, you will get a wrong cross. So you have to write very nicely, with proper handwriting. Capital R, capital Y. Capital R shows round, capital Y shows yellow. Wrinkled, shown by small r. Green, shown by small y. 2 contrasting characters. Now, the parents, they produce the gametes. RY, capital. They produce RY. They segregate as per the first law of dominance. Now, what happens? We have to go for the F2 generation, that is selfing. So, R, capital Y, small y is selfed with the same type of plant. This and this, very important. Then what we do? Third step, we go for the Punett square. Always remember, here you have to draw 16 squares. 16 squares. In monohybrid, there were 4 squares. 16 squares. How? You divide it now. You have to write the alternative, by permutation and combination it is done. How to write? R with big Y, 1. R with small y, 1. Small r with big Y as 1 set. Small r, small y, 1 set. They segregate. Matlab here another law was devised by Mendel, which was known as the law of independent assortment. That's a very important law. I will tell you after this. 2 laws were given in the law in the monohybrid cross: law of dominance and law of segregation. What was given in the law by the dihybrid cross? What did Mendel observe? He observed the law of independent assortment. That is how the gametes are assorting independently. And how will you define it? How is this law defined? When there are 2 pairs of characters, the distribution of the alleles of one character into the gamete is independent of the distribution of the other alleles of the other character. Say this character allele is getting distributed, R small y is independent of R capital Y. Big R Y, big Y, small y, its assortment is independent of this one. Iska assortment is independent of iska. And they are distinct, they are unique. That is the law of independent assortment, a very very important law, and it was given by Dihybrid cross. Okay, so when there are two pairs of characters, like here we have taken round and wrinkled, and one the color of the pod and the texture. When two pairs of alternative characters, the distribution of the alleles of one character into the gametes is independent of the distribution of the alleles of the other character, and as seen here. And this is known as law of independent assortment. And how the gametes are produced. So I think big R, big Y, big R. And what you write here, you will write here also. Make 16 squares first without any confusion. Big R, big Y as one. Big R, small y. Again small r, big Y. Small r, small y. Then cross multiply. Then start doing cross multiplication. I am telling you the technique. Big R, write together. Big Y together. Like this you keep on doing. This, you complete the square. The second slot will be big R, big Y, small y. This one with this one with this one with this. Okay, this is the second slot. Then you, after you complete all, you will see total 16. In this 16, how you find out which is the what type, the phenotypic type, phenotypic ratio. How will you find out? Mendel found out of this dihybrid cross, he found in the F2 generation, the 9 seeds were round and yellow. 9 seeds were round and big R and yellow. Round, the traits, big R, big Y. Both the traits are dominant. So round and yellow. Round is also dominant character, yellow is also dominant character. Represented by capital R, capital Y. So he found in this whole thing, you have to find out how many are round and yellow. He found out 9. First, second, third, fourth, 5, 6, 7, 8, 9. Round and yellow. First phenotypic ratio. Then next three he found out were round and green. 1, 2, 3. Round and green. Another 3 he found as wrinkled and yellow. Wrinkled and yellow means this is a recessive trait. Small r, small y. 1, 2, 3. Wrinkled and yellow. Wrinkled and yellow. 3 upon 16. 3 are there. Okay. And 1 is only wrinkled and green. Wrinkled and green. The last one. Small r, small y is wrinkled and green. Okay. So 3:9:3:3:1, and the total ratio is 16. If you add this, and if they ask you to find out the percentage of the individual one, how much is round and yellow, how much is round and green, how much is wrinkled and yellow, and how much is wrinkled and green, what you will do? Very easy. 9 upon 16 into 100. For the another one, you will do 3 upon 16 into 100. For the another one, again you will do 3 upon 16 into 100. If they ask you, find out the percentage of each of the offspring octet, that is the further question over here. So this I think is clear to you, and very important, Mendel told monohybrid and dihybrid. They ask you an exam, give the difference between the two, draw a Punett square to explain the two. So I have drawn this dihybrid cross to tell you. I think you will also do it nicely when you practice it. You can take any two alternative characters and do it. And this Mendel had, his laws had a wide set of application. It was used for the study of how the offsprings are produced and the possible combinations. What are they? They all were developed, they were done on the basis of Mendel's law. A knowledge of basic Mendelian principles gave us an idea of the new combinations in the progeny of the hybrid, and it enabled us to study their frequency. Second was, it was of great importance for the plant and the animal breeders. It was very much important, okay, for the plant and the animal breeders. And one very important term is there here in your book, last part. What is mutation? If they ask you what is mutation, so you can write a very good simple example of mutation is, it is the sudden change in the genes or the number or the structure of the chromosome. A sudden change in the one of the genes, either in the form of the number or the structure of the chromosome. So it happens on the DNA strand, and that we will study in your higher classes. And this mutation, what are the causes of mutation? The causes of mutation can be the environment. It can be from the birth time, that sudden change, that change can be there, or it can be an acquired trait. Sometimes it can come from the environment. And mutation alters the heredity material of the organism and results in the change of the traits. Now, for example, 2 examples of mutation are sickle cell anemia. That is a blood disease in which the mutation causes the change in the DNA, resulting in the production of sickle cell RBCs. Otherwise, the RBCs are biconcave in shape for absorbing the oxygen. Okay, if you remember the shape of RBC. Here it is sickle cell. That's why sickle cell anemia it is called as. And the next one is due to radioactive radiations alter the structure of the genes, and this also affects the genes, and the alterations are seen there, and it had happened in the Second World War time. Okay, so if I tell you a quick recap of the entire lesson which we had done, and for the part of chromosome, I should tell you there, when I told you sex chromosome, one more word will be there. If they ask you, explain sex chromosome in man, you will explain by that method which I told you. And if they ask you, what do these chromosomes carry? Another question. So what will you write? You will say the chromosomes are the carriers of the genetic material in the form of genes. And these genes, then they can ask you, name a sex-linked disease. I am telling this for the ICSE students. You can come on the page where it is given there, a sex determination, as told you. Then the chromosomes are the carriers of genes, and the genes can be in 2 forms: dominant gene, recessive gene, and then also I told you what is genotype, what is phenotype. Also there is one word here, one word here given, which I want to, you should do it, that table number figure 3.5, where they have shown you that how the genotypes are and the phenotypes are formed in this table, and the dominant and the recessive traits of a human being, like the eye color, the brown is dominant over blue, and the tongue, the different characters are given here. You have to by heart this. And another thing I wanted to tell you here is about the carriers. If they ask you to draw one checkerboard showing the carriers of chromosomes, and draw one simple one. Yes, on page number 30 it is given X-linked inheritance. It is given here. Please come to X-linked inheritance, and the 2 examples are hemophilia, where they are given 2 cases here, case 1 and case 2, very nicely given, and the crisscross inheritance of sex chromosome I have shown you. Based on that, what is a sex-linked inheritance? It is definition you should keep in mind. It is appearance of a trait which is due to the presence of an allele exclusively on the X chromosome or on the Y chromosome, either on the X or on the Y. 2 case studies are given here, one where the mother is the carrier and one where the father is the carrier. The method is same as what I showed you in sex determination of man, where the 50% male and 50% girl is there, and father is responsible for this. So I hope I am clear with this lesson with the LMR, and just on your fingertips will be what is heredity, what is variation, why more variation in sexual reproduction as compared to asexual, what are variants, how they are produced. Then we came to the terms alleles, genome, gene, phenotype, genotype, homozygous, heterozygous, dominant trait, recessive trait. Then we came on sex determination, sex chromosome, autosomes, and the sex chromosome. Then related to that, sex-linked inheritance, sickle cell anemia, given in your book, please see to it. After that I came to Mendel. Who was Mendel? Pea plant, why he selected pea plant, the experimental technique, and his crosses, the monohybrid, the dihybrid. From monohybrid, the 2 laws, the law of dominance, the law of segregation. From dihybrid, he came to the law of independent assortment and the 3 laws. And the end was mutation. So I think it's a very important lesson, and whatever I could convey in this less time, I have done. And please practice this monohybrid and dihybrid cross, and study nicely, and still then stay geeky. Thank you. And and have a nice time and write and practice. Thank you.